Hello friends, welcome to DanTuber. Friends, today we are going to discuss Periodontics MCQs, important MCQs for revision for NEET MDS 2020 preparation. In the previous video, we started with Periodontal Microbiology, and this video is in the continuation of the same. If you have not watched that video, the link is provided in description. Also, if you are able to see the link on the screen, you may go through that video. Otherwise, I have already provided the link in the description. So let us continue with periodontal microbiology MCQs. So the first question is, central gram negative core supporting outer cocal cells are called. So this is a very obvious question. The answer is, it is called corn-cob arrangement. And with respect to corn-cob arrangement, these are highly specific cell-to-cell interactions. Okay, these corn-cob formations have rod-shaped bacterial cells such as Bacterionium and Fusobacterium nucleatum that form the inner core of the structure, and cocal cells such as Streptococci, they will form the attached along the surface of the rod-shaped cells. And supragingival plaque typically demonstrates corn-cob structures. So this is with the explanation part. Next question, breakdown of periodontal fibers in periodontitis is due to bacterial enzyme, collagenase, hyaluronidase, coagulase, none of the above. Correct answer is collagenase. And with respect to collagenase, it is responsible for the breakdown of the periodontal fibers, especially in periodontitis, and it is released by bacteria such as Porphyromonas gingivalis most of the times, and also by the polymorphonuclear leukocytes and some populations of fibroblasts. And the other enzyme given here, hyaluronidase, it causes breakdown of the ground substance and helps in the spread of inflammation. This is also very important point to note down, friend. It causes cellulitis, hyaluronidase. Next question, please. Pellicle formation involves adsorption of acidic glycoproteins from saliva, focal areas of mineralization, focal areas of necrosis, bacterial colonization. So, pellicle formation. The correct answer is adsorption of acidic glycoproteins from saliva. So, with respect to the description part, formation of plaque occurs in 3 phases, as you all know. I think we need to discuss plaque in a separate video. If time permit, I will definitely post a separate video on plaque. So, plaque formation occurs in 3 stages. Formation of pellicle, initial colonization of the tooth surface, and then is the secondary colonization and plaque maturation. And the pellicle formation occurs by selective adsorption of salivary glycoproteins by electrostatic, van der Waals and hydrophobic forces. So this is very important point, friends. So next we will come to which of the following is a common factor for the initiation of both dental caries and periodontal disease. The options are bacterial plaque, lactic acid, calculus, or no common factor. So the correct answer is bacterial plaque, early supragingival plaque, which contains gram-positive cocci and rods, while mature supragingival plaque contains gram-negative rods and filaments, and these organisms cause gingivitis. This subgingival tooth-associated plaque contains gram-positive cocci and rods, which results in root surface caries such as actinomyces viscosus. Then subgingival tissue attached plaque. It contains gram-negative bacteria, mainly Porphyromonas gingivalis bacteroids, which may cause the destruction of periodontal tissue. Next question. Severe alveolar bone loss as observed in juvenile periodontitis is associated with cyclic neutropenia, lysis of neutrophils, increased phagocytosis, impaired neutrophil chemotaxis. The correct answer is D option, impaired neutrophil chemotaxis. So in juvenile periodontitis, there is a basically a functional defect of polymorphonuclear chemotaxis and phagocytosis of the microorganisms. This defect may be induced by the invading bacteria, that is Actinobacillus actinomycetam comitans. The inorganic component of plaque is primarily calcium and fluoride, calcium and sodium, calcium and phosphorus, sodium and calcium. So the correct answer here would be calcium and phosphorus. So basically plaque contains about 5% of the inorganic components, and amongst these the principal inorganic ones are calcium and phosphorus. Apart from these, other elements are mainly magnesium, potassium, sodium and fluoride. And amongst these, fluoride deters the metabolism of the plaque bacteria and aids in the remineralization of the tooth surface. And next slide you will write. And one more thing I would like to discuss here: the source of the inorganic components of this supragingival plaque. It is saliva, and for subgingival plaque, it is GCF. Okay, so this is very important point to note: source of inorganic component of supragingival plaque and subgingival plaque, which is saliva and GCF respectively. Co-aggregation is mainly predominated by interaction between gram-positive bacteria, interaction between gram-negative bacteria, interaction between gram-positive and negative bacteria. D, none of the above. So the correct answer is option B, interaction between gram-negative bacteria. Next question, please. Which of the following is a periodontal pathogen? Streptococcus sanguis, Streptococcus mutans, Actinomyces viscosus, Porphyromonas gingivalis. Correct answer is Porphyromonas gingivalis. Okay, periodontal pathogen is Porphyromonas gingivalis. Amongst these, option C, that is Actinomyces viscosus, it is associated with root surface caries, and Streptococcus mutans is for enamel caries. Increase in steroid hormones are associated with a significant increase in Actinomyces viscosus, Prevotella intermedia, Streptococcus sanguis, Campylobacter rectus. Correct answer is Prevotella intermedia. With regard to this steroid hormones, Corman K. S. et al. In 1982, he concluded that increase in steroid hormones are associated with significant increase in the proportions of Prevotella intermedia in the subgingival plaque. Next question, friends. The carbohydrate not associated with dental plaque is dextrose, levance, glucose, rhamnose. So correct answer is rhamnose, obviously. Specific plaque hypothesis was put forward by Jenko, Listgarten, Losch, none of the above. So the correct answer is Losch. So there are two hypotheses, non-specific hypothesis and alternative or specific hypothesis. The non-specific hypothesis mainly states that the periodontal disease results from elaboration of the noxious products by the entire plaque flora. Okay, so it states that it is by the entire plaque flora. So okay, so the periodontal disease can be treated by debridement, that is surgical or non-surgical, and oral hygiene measures. So this theory has been discarded basically. However, much of our prevailing clinical treatment is still based on non-specific plaque hypothesis. The next one is alternative or specific hypothesis, which states that only certain plaque is pathogenic and recognizes plaque as the pathogenic only when signs of associated disease are present. So this is the what we have discussed just now. And now with regard to Listgarten, the option B, I think, Listergarten described 4 zones in acute necrotizing ulcerative gingivitis. This is also very important. These zones are bacterial zone, neutrophil rich zone, necrotic zone, and zone of spirochetal infiltration. Okay, sir. Okay, friends. Next question. Which of the following is the most numerous component of the plaque? Mineral, food debris, microorganisms, leukocytes. Correct answer is microorganisms. Which surface of tooth has most of the plaque? Gingival third of the tooth surface, incisal 1/3 of the tooth surface, middle 1/3 of the tooth surface, and uniform on all the surfaces. So we have already seen the answer. The answer is gingival third of the tooth surface. So basically plaque forms on the preferentially it forms on the non-self cleansing areas. So plaque will accumulate on the gingival third of the tooth surface. The main reason being the lack of movement and food and tissues during mastication, and also it accumulates in the cracks, pits, and fissures. A bacterial enzyme capable of altering the ground substance of the periodontal ligament is, I think we have discussed in some previous question, amylase, mucinase, dextranase, hyaluronidase. Correct answer is hyaluronidase. It causes the breakdown of the ground substance and helps in the spread of inflammation. In later stages of plaque development, which organism predominates? Cephalococcus, streptococcus, rods and filaments, vibrio and spirochetes. The correct answer is rods and filaments. So let us see. Okay, the description is available. Okay, so friends, this is about today's video. How did you like the video? Please give your valuable feedback in the comment section. Also, if you like the video, please hit the like button, subscribe to the channel, and share it with your friends. Share the video with your friends, share the channel with your friends, those who are preparing for NEET PG or any dental competitive examination. Please share it as much as possible so others can also take the benefit. And also please subscribe, hit the bell icon, and so that you can get the notifications as soon as a new video is posted. So this is all about today's video, friends. Please take care and study hard and study well. Thank you so much, friends.